for external ear spinner, and different conservation methods to prevent species from becoming extinct. Percentage increase is 247, where there are decrease in numbers, reference to increase and then decrease, and then there is a data manipulations, correct comparisons of difference in rate of increase, decrease, providing habitats, protected areas, laws to prevent hunting, education, captive breeding program, use of AI, reintroduction programs. Reintroductions programs, educations, captive breeding programs. This is for conservation methods that are used to increase the number of herbivores, where the risk to the northern white rhinoceros species as a result of its population size is inbreeding, lack of genetic diversity, increased risk of genetic diseases, mutation, reduced ability to survive changes in the environment. Inbreeding, lack of genetic diversity, increased risk of genetic diseases, reduced ability to survive changes in the environment. So inbreeding, lack of genetic diversity, increased risk of genetic diseases, mutation, inbreeding, lack of genetic diversity, increased risk of genetic diseases, mutations, and reduced ability to survive changes in the environment, increased risk of extinction, only small population sets unknown, difficult to locate each other, too old to breed. Inbreeding, lack of genetic diversity, increased risk of genetic diseases, mutation, reduced ability to survive changes in the environment, increased risk of extinctions, only small population sets unknown, difficult to locate each other, too old to breed. Percentage of fish stocks that were sustainable in 2020 is 66%. And overfishing risks to species if its population size decreases, reduction in genetic variation, inbreeding, idea of an increase in the frequency of recessive allies, consequence of having too many recessive allies, risk of extinction, unable to find mates. Reduction in genetic variation, inbreeding, increase in the frequency of recessive allies. Reduction in genetic variations, inbreeding, and increase in frequency of recessive allies, consequence of having too many recessive allies, risk of extinction, idea of unable to find mates, how fish stocks can be managed sustainably, and how overfishing can be prevented by education, monitoring the population's net time mesh size, reduce demand to eat from unsustainable fish stocks, alternative sources of fish, treaties, license, fines is making it illegal, illegal cultures, restricted catch weight, no catch zones, fish farms, cross fishing season, captive breeding and release. Sulfur dioxide, burning fossil fuels, acid rain, carbon dioxide, burning fossil fuels, enhance greenhouse effect, methane, cattle and rice farming, enhance greenhouse effect, fertilizers, crop farming, eutrophication. When fertilizer is applied to fields, what happens in lakes when eutrophication? Fertilizer leach into rivers, producer growth, algal growth, death of producers, increase decomposers, increase respiration, decomposers use up the oxygen in the water, organism, fish creatures die, suffocate, migrate due to lack of oxygen. Organisms die due to the lack of oxygen. Ways in which a farmer could reduce the chances of eutrophications: use less amount of fertilizer, use slow-release fertilizers, do not apply after rain, do not use near water, make channels between land and water body. Do not use near water, and only apply when crops are growing. Increase decompositions, increase respiration, use of oxygen, seawater organisms die due to lack of oxygen. To increase crop yield production, to reduce competition with weeds. Increase crop production and to reduce competition with weeds. How herbicides kills water plants, lack of producers, food for herbivores. Bioaccumulation, reduce biodiversity, lack of roots cause erosions. Kills water plants, lack of producers, bioaccumulation, reduce biodiversity, lack of roots cause erosion. Network branch leaf, network branch veins, broadleaf petiole. Network branch veins, broadleaf petioles. So the plant hormone is auxin, and herbicide kills plants, no chlorophyll synthesis, cannot trap sunlight, cannot photosynthesis. Network branch veins. Network branch veins and broadleaf and petiole. Broadleaf petiole. Network branch veins and broadleaf petiole. Bacteria type of pathogen penicillin used to treat antibiotic name of group of medicinal drugs that includes penicillin and nucleus, mitochondria, vacuole. Features of fungus cells used to distinguish them from cells of prokaryotes: nucleus, mitochondria, rough ER, vacuole, hyphae, structure of cell wall. Nucleus, mitochondria, rough ER, vacuole, and hyphae. Nucleus, mitochondria, rough ER, vacuole, hyphae, structure of cell wall. Genus name penicillium. Two nutrients that need to be added to a fermenter to produce penicillin is glucose, amino acid, salts, ions, water. Glucose, amino acid, salts, water. Nutrients are sterilized before they are added to the fermenter. It stops the growth of microorganisms and prevent contamination. Stops the growth of microorganisms, prevent contamination. Fermenter is a gas outflow pipe because it is release of waste gas, carbon dioxide. Carbon dioxide gas is produced during fermentation to prevent fermenter exploding. To reduce pressure, carbon dioxide is toxic to fungus if it builds up. Step 4 is reproduction of A before adding to fermenter. And Step 5 is inoculations in the fermenter, and Step 6 is the exponential growth of organisms A in the fermenter. Exponential growth of organism A in the fermenter. Exponential growth of organisms A in the fermenter. Step 6 is exponential growth of organisms A in fermenter. And sampling to check when penicillin is ready. And penicillin is a byproduct of maximum growth or toxic fermentation. Penicillin is a byproduct of maximum growth or toxic fermentations, and cells penicillin are removed and purification, filtering, packaging of penicillin. Bacteria are useful because small cells do not need much space. There is no ethical constancy. It can form a very complex molecules. It shares the same genetic code with us, and bacteria are they only need a simple requirements. They don't need much space. Rapid reproduction rate. And they have plasmids. The organ that contains the cells that have been destroyed is pancreas. Type 1 diabetes caused by destruction of the cells. Reduced blood sugar concentration. Lag exponential. Stationary death. Limiting factors that limit not enough space, overpopulation, competition, fewer nutrients, not enough oxygen. Limiting factors. Limiting factors, not enough space, and competitions, and there is fewer nutrients, there is not enough oxygen. There is a change in pH or change in acidity, and there is an increase in temperature. Factors that limit not enough space, competition, fewer nutrients, not enough oxygen, change in pH, increase in temperature, build up of toxic waste substance. to make hemoglobin, to make red blood cells for transport of oxygen, iron deficiency anemia. Restriction enzyme shortening of enzyme that is used to cut out the gene from their DNA. How the gene form recombinant DNA? They use the same restriction enzyme to cut the DNA of the plasmid, and then the plasmid of the bacteria, and then their gene from the DNA, and it's complementary. They form sticky ends, and the sticky ends must be complementary to each other, and then it's dried by DNA ligands. So it uses same restriction enzyme to cut their cassava DNA, and sticky ends are formed, so complementary ends joined by ligands. Complementary ends joined by ligands. Same restrictions enzyme to cut cassava DNA, sticky ends form complementary ends joined by ligands. Scientists develop genetically engineered varieties of crop plants, often breed them for just several generations before releasing them for farmers to use. So, ideally they need to check that offspring inherit new genes, or ensure new variety can grow in field conditions, make sure the new gene does not cause any adverse effects, make sure the GM plants have the same and high enough yield. Ideal building up a large quantity of seeds to sell to farmer. Need to check that offspring inherit new gene, ensure new GM variety can grow in field conditions, make sure the new gene does not cause any adverse effects. Plants have this high enough yield, building up a large quantity of seed plants to sell to farmers. Cell membrane, cytoplasm, ribosomes, DNA. Cell membrane, cytoplasm, ribosomes, and DNA. Rapid reproduction rate reproduce asexually, small do not need much space, simple requirements to grow, ability to make complex molecules, few ethical concerns, and presence of plasmid same genetic codes in other organisms, great variety of bacteria could be useful in biotech. Ability to make complex molecules, simple requirements to grow. So there is a death praise because limited lack of resources, increased competition for resources, and build up high concentrations of waste, toxins, and build up of pressure, increase in temperature above optimum. So there's a death praise where the death rate above birth rate, limited resources, increased competition for resources, build up of waste, build up of pressure, increase in temperature. Sufficient kinetic energy and optimal temperature for many collisions between enzyme and the substrate to maximize the rate of reaction, to prevent enzymes at the site from denaturing at higher temperature so that at the site does not change shape. At optimal temperature at the site is complementary to substrate reactants. Amino acids are used to make proteins quicker, efficient absorption. Amino acids are soluble, dissolved. Make proteins quicker absorption, soluble, dissolved. cytoskeleton, dendrites, and spermatozoa. Animals have 3 pairs of legs, wings, head, thorax, abdomen. No pincers, carapace. Animals pincers of different sizes, eyes on stars, smooth carapace, has 5 sizes, angular, have blisters on legs. 4 pairs of legs, fierce cephalothorax, pedipalps. Inherited structure. Inherited feature that functional feature, inherited structure and functional feature that increase the fitness, the chance of survival, chance of reproducing to relieve offspring, camouflage, warning signs, gas predators. Identifications can be done using base sequence pattern in DNA genes. Base sequence in DNA. And each species has unique DNA compared with the base sequence of a known species. If a match with DNA from the known species, then the DNA is from that species, the base sequence, and any turning involved in DNA analysis. Reasons an animal species become endangered: hunting, pollution, pesticides, insecticides that kill animals. Hunting, pollution, insecticides that kills animals, and loss of habitat, and disease, and increased competitions, and increase in predators, and climate change, and lack of food. So reasons why animal species become endangered is hunting, pesticides, insecticide, and loss of habitat, disease, increased competitions, increasing predators, climate change, lack of food, loss of prey. Hunting, so hunting, pesticides, lack of food, loss of prey, increase in competitions, and there's a habitat destruction, there's a climate change, there's war, there is increase other disease, and increase in predators. Similarities and difference between a parasite mesophyll cell and a bacterial cell. Similarities and difference between a parasite mesophyll cells and the bacterial cell. Presence of cell wall, cell membrane, DNA, genetic material, ribosomes, cytoplasm. Cell membrane, DNA, genetic material, ribosome, cytoplasm. Difference: no plasmid, no flagella, no capture, no loop of DNA. Presence of chloroplast, presence of mitochondria, presence of nucleus, presence of large vacuole. Chloroplast, mitochondria, nucleus, large vacuole. Chloroplast, mitochondria, nucleus, large vacuole. Bacteria release a toxin causing chloride ions into small intestine, reducing water potential in the gut. Osmosis and water move into the gut, into the intestine, out of the cells. Larger secure area has prevented most growth, killed most bacteria. Bacteria is no resistance to antibiotics. How bacteria become resistant? Natural selection, mutation. Bacteria without resistance die. Bacteria with resistance survive. Resistant bacteria multiply, passing the genes for resistance onto the offspring. Antibiotics destroy the bacterial cell walls. Resistance pass through the plasmid. Resistance pass through the plasmid. Passing alerts genes for resistance onto the offspring. Passing the genes for resistance onto the offspring, resistant bacteria multiply. Only take antibiotics when needed. Complete the course of treatment. Restrict the use of antibiotic B. Communities are the populations of different species in an ecosystem. Of the populations of different species in an ecosystem. Of the populations of different species in an ecosystem. So adaptation is sometimes defined as the process resulting from natural selection by which populations become more suited to the environment over many generations. Range as turns downward, range as turns upwards to higher altitude. Range does not extend downwards much. And little change in range. So decrease in populations of island trouts, competitions between species at higher altitude. Reasons for the change in the range of island trouts is decreasing populations of island trouts, competition between species at higher altitudes, and increasing populations. There is competition with other species for breeding nesting sites at higher altitude, habitat loss, deforestation, disease, predation, loss of food source, climate change, increased hunting at lower altitudes, increased pollutions at lower altitudes, introductions of new species, natural disasters. Natural disasters, competitions, and they're usually more about habitat loss, deforestation, climate change, predations, and overhunting. There's diseases, natural disasters, overhunting, and loss of food source predation. Pollution, introduction of new species. The risk of these species of birds that have decreasing populations, difficulty finding a mate, and inbreeding, reduced fitness, difficulty finding a mate in breeding, reduced fitness, decrease in genetic variation within each population, increase in genetic disease, more vulnerable to pathogenic diseases, unable to adapt to changing environment, increased risk of extinction as populations are small by natural catastrophe, introduced species, have evolved different shape beads is natural selection, variation in bead shapes, and mutations, and those birds with slightly advantageous unique bead shapes more likely to find food and survive. Natural selection, variation in bead shapes, mutations. Those birds with selective advantage, unique beak shapes are more likely to find food and survive. So the birds that survive and reproduce, personally alliance continues over many generations. Substance in red blood cells that combine with oxygen is hemoglobin, and name of component of blood that promotes blood clotting is platelets. Salt solutions has a lower water potential than the red blood cells, so diameter decreases because water leaves the red blood cells by osmosis. Water travel from higher water potential to lower water potential down a water potential gradients across the partially permeable membrane. Across the partially permeable membrane. Water travels from a higher water potential to lower water potential. There is no change in the mean diameter of another one because water potential of red blood cell is the same as the salt solution, so there's no overall movement of water across membrane. Red blood cell burst when immersed in pure water, but plant cells do not, is because red blood cells do not have a rigid cell wall. Red blood cells burst. Red blood cells burst when immersed in pure water, but plant cells do not. It's because red blood cells do not have a rigid cell wall. The uses of water in a plant is transport of sucrose translocation as a reactant used in photosynthesis as a solvent dissolved in it, medium for chemical reactions, and there is support maintaining turgidity of cells, prevent wilting, cooling, temperature regulation, germination, elongation of cells, and growth. As a solvent. Potato cubes loses greater percentage mass. Movement of water out of water is caused of mass loss. Water moves from an area of high water potential to an area of low water potential. Water potential of that solution is lower than water potential of another solution. Greater stable water potential gradients in one than another. Relatively more water leaves the potato cubes. So, as better appearance of a cell from a potato cube that has been immersed in the distilled water for 30 minutes is, it will become swollen, larger, bigger, cell is turgid, vacuole is swollen, cell wall bulges, cell membrane, cell contents, process on cell wall. Active transport use energy from respiration. Use energy from respiration. Transport is against concentration gradient. Involve movement of iron substances other than water. Involve protein carriers. Root head cells. Leaves are yellow. Leaves show chlorosis, and medicine required for making chlorophyll. The effect of a lack of magnesium ions on the color of plant leaves is leaves are yellow and magnesium required for making chlorophyll. So, featured movement of particles always occurs across our cell membrane. Molecules of movement of molecules during gas exchange. Requires energy for respirations and rate of movement of particles is higher concentration. Gradient is larger movement of particles during gas exchange. Occurs across the cell membrane. Active transport for ion uptake absorption. Ion concentration lower in the soil than in the root cell. Movement against the concentration gradient creates a water potential gradient, like example of ion like nitrate medicine. Blue at time 0 indicates low glucose present and shows low glucose on the outer surface of dialysis tubing. Water is a result of error. Green yellow red indicates presence of glucose. Glucose move out of dialysis tubing to water. Movement without the concentration gradient. Dialysis tubing is permeable to glucose. Glucose diffuses out of dialysis tubing movement without the concentration gradient. So, either the Benedict solution changes color quicker, give more intense color. Benedict solution changes color quicker, give more intense color. Air microbiome allows movement of substance into the cell, increase surface area for absorption by diffusion or active transport, and have proteins in the membrane for active transport. Allows movement of substance in the cell, increase surface area for absorption by diffusion, have proteins in the membrane for active transport, and rough endoplasmic reticulum is site of protein synthesis. Modify proteins assemble amino acids in a specific sequence to make lean proteins. Site of protein synthesis, modify proteins, assemble amino acids in a specific sequence to make lean protein. Mitochondrial aerobic respiration provides energy for lean cell process. Corolla and loss of water by osmosis downward potential gradient, diarrhea, dehydration, loss of sorts. Loss of water by osmosis and diarrhea, dehydration, and loss of salts.